Hello students, welcome to lab number 12. We're going to be talking about mitosis. Basic structure of today's lab assignment. You're going to be going through most of today's lab assignment, and most of it is going to be drawing out and labeling most of the pictures and doing most of the tables. Okay, so I've posted that online. All right, introductions. All plants and animals on the planet grow during their lifetime. This is a process in single cell dividing, producing two identical copies, called mitosis. Before a cell can divide, all DNA within the nucleus of the cell must be replicated. If this does not occur, the newly created cells would not contain the correct number of chromosomes, and genetic information would be lost. Mitosis occurs in phases. These phases of mitosis, the cell spends a lot of time at rest. However, the cell does not really resting. It's growing. Right, it's becoming physically larger. The DNA is replicating and the cell is getting ready for division. So it's becoming larger, DNA is replicating inside, and it's getting ready for that division by replicating the organelles, replicating the mitochondria and chloroplasts, if it has it. Okay, during DNA replication, this occurs during interphase, the S phase, you remember. The duplicated chromosomes contain two chromatids held together at the centromere. That's that center part. These chromatids are called sister chromatids. At the beginning of interphase, within the nucleus, there are a total of 46 chromosomes in us, right? At the end of interphase, there's still 46. However, all the genetic material being copied, there are 92 chromatids, right? There's still 46 chromosomes. There's just 92 chromatids, right? So they're duplicated. There's 92 chromatids. There's still 46 chromosomes. During interphase, the cell grows and gets ready to divide, right? So we have G1, S, and G2. The cell spends approximately 18 to 23 hours in interphase for us, right? So G1, we have some gap, that's the cell growing, S, DNA synthesis, and G2, we have a little bit more growing. The first phase is G1, that's gap 1, right? During G1, the cell makes sure it has all the enzymes available, right? So the cell grows, becomes larger, copies all its organelles. S phase is DNA synthesis phase, or synthetic phase. One replicated chromosome, you have a centromere, and then you have sister chromatids. Two sister chromatids per chromosome, so all of it is a chromosome, you have two sister chromatids. The last subphase is G2. So this is the final stage of cell growth. The centrosome containing centrioles is also replicated. So you have the centrosome. This is where the spindle fibers will be produced. Right, and so the spindle fibers will go down and attach onto the centromeres. So before we head on to mitosis, you have to get into that checkpoint, right? So in order for the cell cycle to proceed on to the next phase, you have to get past the checkpoints. Remember, we talked about this during lecture. So G1 checkpoint, before you get into S even, you have to get through that G1 checkpoint. So that's a restriction point, determines if conditions are favorable for cellular division, for nuclear division. So if this checkpoint is not passed, it goes into G0, right? So it's not going to replicate the DNA unless it gets past that point. And this is usually if it doesn't have enough nutrients, if it doesn't have enough nucleotides in the cell in order to replicate the DNA. Usually at this point, there are some cells that don't actually go through the S phase, some cells that don't actually go through and replicate the DNA. Some of these cells are muscle cells, brain cells, nerve cells. They don't actually replicate the DNA. They don't need to, right? So they just sit there and they live, basically. Okay. Then there's G2 checkpoint. After the DNA is replicated, there's a bunch of enzymes, a bunch of proteins that go through, make sure it has been replicated. So this is a G2 checkpoint. After G2, then comes mitosis. After a certain amount of mitosis, then you have metaphase checkpoint. If the chromosomes have been attached to the spindle fibers. If those chromosomes are attached, if the spindle fibers have been attached to the centromeres, then we can move on to anaphase. So, after metaphase, that checkpoint says that the chromosomes can be, or the sister chromatids can be split apart, and then the cells can then go into cytokinesis. All right, now we can talk about the phases of mitosis. We've already talked about this during lectures, so I'm just gonna quickly and briefly go over the different phases. So prophase, this is where the chromatids begin to condense into highly compacted structures, right? Where they start to wind into these structures. The nuclear envelope starts to break down. Spindle fibers are formed, but they don't go into the nuclear membrane yet. And they can't be attached to the chromosomes yet. After this phase, we have prometaphase. The nuclear envelope is completely broken down, and the spindle fiber is fully formed. The nuclear envelope is gone, and then the spindle fibers can attach to the kinetochores, right? So they can attach to those centromeres. Next, we have metaphase. Metaphase is where the sister chromatids can line into the center. Remember why they need to line into the center. Because then they can split apart evenly. If they didn't line up into the center, some parts would go into one cell more than other. And then once we get past that checkpoint, we can get into anaphase. Anaphase is when they start separating. When they start separating, the individual daughter cells, the individual sister chromatids become daughter chromosomes. And then we get into telophase. Telophase is when they've reached the respective poles. Those daughter chromosomes have reached their respective poles and they begin to decondense. They begin to unravel. The nuclear envelope reforms with two polar separate nuclei. Cytokinesis is formed usually during anaphase. In animal cells, it forms a cleavage furrow. In plant cells, it forms a cell plate. These are different phases of mitosis. If you can identify these phases and put them into the correct phases, the correct order and phases, then you should be able to know what I might ask you on a quiz, what I might ask you on a test. So, different slides down, we can go through these. I'll just wait. Okay, so E down here is interphase. All chromatids in DNA are replicated here. This is what interphase would look like. D right here, this is prophase. This is what the chromatids would look like if they were condensing. This is prometaphase, B. The nucleus is completely fragmented here. And you can see the spindle fibers coming into the nuclear envelope. F right here is metaphase. You can see the chromosomes in the center here. The chromatids are aligning. C right here is anaphase. You can see the chromatids separating apart into daughter chromosomes. And then A is telophase, and you can see the cytokinesis forming that furrow. Okay. This slide represents a real life slide of a plant cells. And I would like for you to look through these and see how many you can find by yourself. How many different phases you can find by yourself. And you can pause the video and you can see how many phases you can see. And the next slide will show you different examples of what some of the answers would be. Okay. Here's some questions for the lab practical. What I might ask you about this chapter. The next chapter is meiosis, and then after that we have our second lab practical coming up on the 28th. All right, so make sure that you're studying for that. All right, I will see you later.